Good morning, one and all. As well as maybe good afternoon also. So, as planned, as of now, I would finish my session, and second session should be happen. So, due to the time constraints and those things and all, now I'll start up the session. Once my session is over, we'll go for the lunch. And afternoon, we'll try to finish off all the sessions what we have planned. Or else tomorrow morning, we'll make it as an earlier one. So, lunch time will be 40 minutes only, and lunch will be provided in the first floor. Chemistry wing. We have made the arrangement in there only. So now I'll start with the computational and overview of the computational drug discovery, because we are going to discuss about the drug discovery, right? So first, I'm just going to give the very basic preambles. So what are the things we are going to discuss in the next 2 days? So as usual, this presentation is only for education purposes, and all the rights goes to their rightful owners, because some images, some data which maybe I have taken from published articles and other things and all. So it belongs to that things and all. So first, I should want to discuss about the word drug. Drug is what we call. It can be either a particle which is intended for the purpose of either the diagnosis, either the treatment, either the mitigation, or the prevention of any disease in man or animals. So the source of drugs may be either the chemical sources will be there, biological sources will be there, so many things will be there. What are the sources of the drugs? If you look into that, we have various sources. One from animal sources, plant sources, rather than that inorganic materials, and the synthetic one we can able to see. So plant sources will play a major role, as India is the biggest boon of medicinal plants, and many drugs have been identified through the support of from the plant sources only. If you take the drug discovery process in general for a new disease or a new target, if they are done, so they will start with the screening of natural products only, because from natural products they have the potential to prove that it can able to inhibit that particular mechanism. That plays a major role. So what happens? Why so much of drug is not identified from the plant or directly marketed, in the sense they may have toxicity, where this will be called as lead identification process in drug discovery. Once I have identified a lead from a plant sources, that I'll take as a medicinal chemistry point of view, will be changing the function group to reduce the toxicity and to improvise the pharmacological profile. So that's a major reason. Example, if you look into the pharmacognosy point of view, quinine sulfate directly came, artemisinin directly came. So such kind of drugs which showed potency during the animal studies or human studies, it have been utilized directly. But some which couldn't able to make it out, there it may have been converted as a chemical thing, synthetic thing. Either it can be completely chemical, it can be a biological penicillin from penicillin notatum, and biotechnology insulin. These are the various sources of enzyme. And what is the need for new drugs? One is unmet medical need. We are facing day by day new diseases, new conditions we are facing. Example, long back we are faced with the COVID. Now Nipah virus, monkeypox virus, so many diseases are coming on. To overcome that, we need to identify medicine. Still for COVID, there is no medications. Only the symptomatic treatment we are happening. So such kind of thing. And you look for the efficacy point of view, low efficacy. Many diseases where still we couldn't able to find a potent drug. Example, if you look for the dementia or Alzheimer's, as a world leading disease of number 5, where still only 4 drugs have been approved, 5 drugs have been approved. In that, 1 drug have been withdrawn from the market also. But it's one of the world leading diseases. So we need to find a drug. And if you come for the side effects of drugs, if you see any what do we call leaflet of any drug boxes, there you can able to see the word called a side effects. To overcome that, some basic side effects are there. As in a pharmacological point of view, will say no. What is the side effect? Nausea, vomiting, diarrhea. The same thing. Everywhere it will be present. Why? Because that kind of side effect happens. Our biological system has n number of enzymes, n number of proteins, which may have the similarity in nature. So that similarity, the drug may get confused whether I should bind with this protein or this protein. So due to that, we are facing side effects and all. So to overcome the side effects, we have to find out a medication. That makes sense. So, and the next one is the cost of the therapy. If you look for the cancer therapy and such kind of things, some injection may cost more than lakhs, 10 lakhs, such kind of things, where which cannot be afforded by middle class or upper middle class or lower classes. So to overcome that, the less effective, we need to find a drug and to sustain industrial activity. As you know about the patent process, so once the patent is over, anyone can able to get on medications. Example, a company A will be there. That company A will be working to find out a new medicine as a monopoly. Like what do we call? This medical business is a big mafia business only, where they want to be the one and only solo leader, where they don't want the others to grow. In the sense, example, let be a company A. That company A will be working to identify new medications, where they will be having success and failures. Success may be at the moment they may get it, but failures will be present, right? That failures how to overcome, because phase 1 it may fail, phase 2 it may fail, or during the process. That amount all together they will be invested to get a new molecule. So once the molecule is prominent, they will be selling. So example, the company will be selling that for a 1,000 rupees per strip. That is patented. They will be selling their name, the branded name A. So B company is there where they will go and get the process permission, like that this molecule we will be utilizing. So they will be paying this what to be called royalty kind of money to the company, and they'll get back 1,000 rupees, and you'll be selling for 500 or 800 something else, as you have not invested much. After some quite period, some 5 years, 8 years, because patent has its own validity. So 10 years, once it is patent is broken, it is ended, in the sense anyone can able to what do we call synthesize or market the product. So I'm the company X, and the company not A, B, and the company X. I'm the last person to be the company. What I'll do is I'll be trying to procure the basic materials from China. So where you will be selling all the AP products is the biggest exporter of AP. So very less quantity. In India, it will be more cost. There, more quantity, less cost. So based on that, I'll be importing the drugs and I'll be making a manufacturing. So once I have done the manufacturing, I'll be selling that medication for 10 strips. Or one strips, I'll be selling for 100 rupees. One person selling for 1,000, someone is selling for 800, someone for 500. I'm selling for 100 rupees. So if you go to a medical shop and if you ask that, I need this prescription, what the pharmacist will say, this medicine is not available in this name, some other name is available, can I give? As we, even though we educated, will be asking question, is there any difference? Oh, no, same molecule will be saying the word. So then what is the difference? This is 1,000 rupees, this is 100 rupees. Usually what I'll do, I'll go for the 100 rupees, cost effective. So due to that, the production or the cost of the companies will get slowly get down. So that company, again, they will try to become a monopoly. They want to win the market. They want to rule the market. So if you have seen the whatever Nifty and those indexes and all, sometimes one company will be big, some companies will be changing. The reason behind that is, this is the reason. So to overcome that, we just require. And more importantly, the last one is the drug resistance. We are facing heavily. We are facing the drug resistance heavily. Why? Because we are not following the protocol as prescribed by the doctor, or how the clinical pharmacists are giving the counseling. We are not at all obeying their words. Example, I'm the person. I have a cold cough. Even though I'm a pharmacist, I won't I won't go to the doctor. I'll go to a local medical shop. Bro, I'm not well. He may try to counsel. No, you have to take 6 tablets, or 6 times you have to take as a course of period. What I'll say, 3 times sufficient. You want to earn, for that you are asking money from me. I'm not ready to give, like that I'll say and come. Inside my body, the organism which is present will never die completely. It will be present, at least some 20% or 30% will be there. Then again production will go off. Again I'll get again and again. So at last, what it happens in the sense, it will become resistant. What happens is a thin biofilm will form quorum sensing. There is a term called as quorum sensing, which is playing a role in resistance process. It knows, okay, something is coming, I should cover up. So once it is cover up, I can't able to do anything, right? Example, how this can be explained in the sense, we have a mobile phone where everyone have the fingerprint to open. Directly, if I keep the fingerprint, my mobile will get unlocked, right? In case if I keep a thin film and if I try to keep my fingerprint, it will never open, right? The same way, the drug which I'm going to take will never enter into the cell wall of the bacteria or the organism which is affecting me. So that we need to find out a new medication. Drug resistance, you know about the methicillin resistance, Staphylococcus aureus resistance. So many things are playing a role. So in general, drug discovery process will take place in almost 15 years of it will be taking time. First, we'll be identifying what is the protein involved, which is the responsible receptor is involved in the particular cause of a disease or particular disorder condition. Then I'll be going to find out a new drug using so many methods, and then I'll go for animal studies, clinical, that is preclinical studies. Then I'll make a suitable formulation. That also plays a major role, because what type of disease I'm going to work on. Example, if I take the paracetamol is available in tablet form, suspension, syrups, injection, parenteral, all forms available, because to deliver that particular drug, it needs. So which type of disease we are going to? So all these things, if I have done and I have our data properly, I should go for filing an IND, investigation of new drug. Once that the investigation is done, then I'll go for human clinical trials. It will take almost some 2 to 10 years. So once this is done, I'll go for a NDA, new drug approval. I'll make it up. So once that is done, I'll be marketing, post-marketing surveillance. This is the general routine drug discovery process which takes place in the earlier stages. So what are the complexities in the sense it have so many complexities because one drug, as I said, right, it should reach a particular site of action. So it has to pass all the things, and that with a valid medical hypothesis, if I have a 10,000 drug candidates, 10,000 all are drug candidates, then it has to pass all the filters. Like how we can say in the sense, in Amazon or Flipkart or some any other what do we call in the search engines. If I want to purchase a mobile, I can keep a mobile. What is the cost? What is the RAM size? What is the space, camera clarity, like that? No, it has to pass all the things, and it should at the last it should go as a one single molecule should come. So if you look, it should be targeted, selective in nature, potent, physically stable, soluble. Why targeted in the sense which region I have to work on? Selective, because we have the adrenaline receptors, right? In heart also we have, in lung also we have. By mistake, the drug which I am taking for the heart disease, if it goes and attacks the lung, I'll get the ticket. Or the drug which I have to act on the lung, if I go and attack my heart, blah. So all things has to be managed, physically stable, soluble, permeable, and more important, it should be patentable. If it is not patentable, there is no use. Not even a single paper should come out as a someone's paper. And most importantly, manufacturable. Why? Because in a bulk manufacturing, they will be making a sensor. In labs and all, we'll be making small beakers or small RBF and we'll be making 1 gram, 2 gram and all. But for the company process and all, we need more amount of quantity. So for that, all things should be considered, and at last only a molecule will try to come out. How this modern era, how the computers are supporting in drug discovery process. So if you look into that, now we are in the omics era, OMICS, genomics, proteomics, metabolomics, lipidomics, studying specifically about a gene is called a genomics. Studying only about the protein is called as proteomics. Lipids, lipidomics, metabolites, metabolomics, such kind of things are there, where this will help us to identify a personalized target that I can able to hit that so that the disease condition can able to calm down. So, or the receptor condition can be balanced. That's the purpose. And then high throughput screening, and that also now outdated. We are not at all carrying out high throughput screening. Now we are carrying out VHTS. High throughput screening in the sense, if I have 1,000 molecules, that all 1,000 molecules I'll take, I'll go for lab assays, in vitro assays I'll be doing, and from that I'll identify. Now though we are what do we call? We are overcoming that those kind of things, and why? Because a lot of chemicals are used, a lot of time is taking. So to avoid such kind of things, we are using these things. And next, if you come the virtual screen, that plays a major role. What we are going to discuss in the next 2 days, that will place the things. One about the and then molecular modeling, these things and all. These are the precepts which will be helpful in reducing the drug tox process. And moreover, even the if you take a drug, it will be of two parts. One is pharmacodynamic and another one is pharmacokinetic. So through molecular docking studies, you can able to come to know the dynamic property. The same way, if you carry out the ADME study, then it will go with the pharmacokinetic property. That also will be discussing. So what is the CADD? You are saying the computational tool, something and all, right? So I should define no. So computational tools or CADD, it's a mathematical approximation. Its major goal is a mathematical approximation and computer programs with the help of maths. We know that, right? Algebra, matrix, such kind of differential calculus, such kind of things we have studied, right? Same way here, everything as an equation with the help of computer programs or algorithm. If I upload a chemical structure here, or if I upload some smileys, what it will do? It will predict and say either this component or this compound will produce a biological activity or not. Future drug or it won't be a thing. So whose major goals are to create efficient mathematical approximation and computer programs that can calculate the properties of future drug molecules and thus helping the process of drug design and discovery. So this is how it will take place. And what is the need? As I said, right, if I'm going through the high throughput screening or something else, if I'm making out, I need what do we call? More amount of time, more amount of chemicals will be wasted. Even the environmental hazards also will be there. So to overcome that, we will be utilizing the computer drug design models. So as you know, in the general term, in vitro and in vivo. In vitro means outside the body, in vivo means inside the body. The same terms will be utilized in computers. That is in silico, we'll be calling them. Why in vitro, why in vivo have been made as italics, right? They are the studies to be carried out to evaluate a chemical compounds or the phytochemicals, what are the thing may be, whether it can produce a biological activity or not. So the same way here, using the computers, we'll be predicting the activity of a molecule, or we'll be carrying out the drug design studies. So that the studies which will fall under the computational studies will call as in silico, and the same way we have tried in the italics mode only. So in silico drug design, and moreover we are in the silicon city of Bangalore. More amount of computers are used, right? And the computers are made up of silicon connectivity, silicon dots, that keyboards and all, the what do we call motherboard, chipboards and all. So that's why it is called as in silico studies. So this is the Bible of computer drug design. What type of way I have to make it out? How I can able to go for a study? What type of data I have? What type of things I need? So based on that, two things are required for a study. One is I need a protein, and another one is I need a ligand. So if I know the protein, what I can do? If I know the ligand, what I can do? If I know both, what I can do? If I don't know both, what I can do? So it will give the plan or the sketch of the study. So if I know the protein structure and if I know the ligand structure, the study can pass under this category called as structure-based drug design. There are three drug design studies. One is structure-based, ligand-based, fragment-based, FBDD, LBDD, SBDD. These are the terms which will be utilized in drug design process. Structure-based drug design. If I have the protein structure, I know the protein structure, and if I know the ligand structure which I am going to work on, then it will fall under the category called as structure-based drug design. This will be in general protein ligand docking or protein docking will go up. This will be this process, how I have to carry out a molecular docking studies, how to choose a protein. Those things will be explained in the upcoming sessions. So if I know only the protein structure and if I don't know what type of ligand will work on, then that the study is called as de novo drug design, fragment-based drug design. I should start fresh. So, and in case if I know the protein structure, sorry, if I don't know the protein structure, or if even though I know the protein structure, I won't bother about that, in the sense it will fall under the category as LBDD, ligand-based drug design, where QSAR modeling will come, pharmacophore modeling will come. And if I don't know the protein structure, if I don't know the ligand structure, then there is of no use with the CADD. So this is the outline how I'm going to work on structure-based drug design, ligand-based drug design, fragment-based drug design. So in the next 2 days, we are going to discuss only majorly SBDD and LBDD only. We are not going to discuss about FBDD. So de novo drug design. First thing, but I want to put an outline about this thing. So de novo, in the sense, in term, we can able to say as a fresh starter. Freshly, I'm starting. Why where this kind of study will be utilized, in the sense like now we are facing with monkeypox and Nipah viruses and all. So where those structures for those kind of diseases, we don't have new drug molecule or what type of molecule to work on. So as a biologist or as a, what do we call informatician, bioinformatician, chemical, whatever the thing may be, what I'll do is I'll take the protein structure and I'll go for modeling. That is either if I know the protein structure exactly, I'll take the extracurricular. If I'm not, based on the sequence, I'll be taking the homology modeling. Homology modeling will be discussed by in the upcoming session by Samira. So we'll be discussing how to prepare the protein, how to model the protein. That will be discussed. So once I model the protein, I have to find out an active site. When the active site is identified, based on the active packet, based on the active packet, I'll be trying to fragment. I'll chop my ligand structures. Here, if for example, if it's a donor, if it's an acceptor, if it's a hydrogen, but what will be aromatic compound. So I'll place one by one, and then I'll be linking them one by one. So fragments, I'll make it out and I'll fit them. Oh, here I require a donor, here I require an acceptor region, and this much distance, how I can fill. So based on that, if I design a work, then it will fall under the category called as de novo drug design or fragment-based drug design. So here, this will give a preamble that this kind of compounds can able to bind with that particular protein. So here the softwares available are Grow, Legend, Ludi, such kind of softwares are available, where fragments will be pre-built library will be there, so that they can able to incorporate into it, and you can able to design some compounds. Next one is pharmacophore modeling. This pharmacophore modeling will be discussed by our scholar Mazin Khan. He'll be discussing how I can able to make it out. So pharmacophore modeling. What do you mean by the term pharmacophore? If you take a drug structure, if you take any chemical, it can be of two parts. One is pharmacophore, another one is oxophore. Pharmacophore means which is responsible for producing the biological activity, where a pharmacodynamic property will play on the pharmacophore. And pharmacokinetic property, how it will be the oxophores, where the additional function groups will be bounded that will helpful in physiochemical properties, improvising the physiochemical properties, where it will be required everywhere. Example, if I have to target my brain, my drug should pass the blood-brain barrier. For that, my log P value should be increased. So that is required, right? So such kind of property will be oxophore. Now I'll come back to the pharmacophore. So pharmacophore modeling, if I take there are two ways of doing pharmacophore modeling. One is based on structure-based pharmacophore modeling, and another one is ligand-based pharmacophore modeling. Structure-based pharmacophore modeling, all the studies of what do we call biological studies or the computational is what we are going to make it out. It all should be in 3D models because our biological system is of a 3D model. Structure-based pharmacophore modeling and ligand-based pharmacophore modeling. Structure-based pharmacophore modeling. What happens here in the sense, a protein structure will be there, a molecule which already have bounded and produces the biological activity. So we'll be taking the 3D structure in which pose it are bounded, what is the functional group, what type of interaction it made. So based on that, if I go for a similarity search, I'll get an idea, I'll get a list of compounds, that is called as a structure-based pharmacophore modeling. Another one is ligand-based pharmacophore modeling. Example, if you have studied in your pharmacological classification point of view, for one disease, one mechanism, 5 to 8 drugs will be acting, where all the 5 to 8 drugs will never be of a seamless structure. All will be of a different scaffolds. So those scaffolds, what happens there? One will be benzimidazole derivative, one will be what do we call quinoline derivative, cenolin derivative, but all together will never be similar. What I'll do is I'll try to superimpose all the chemical structure in a 3D pose. This is 2D, this is 3D, because dihedral angle, torsion angle, all the angles will be playing a major role. So that's make the sense, because body requires the bend, the rigidity, the flexibility, all plays a role for a binding. So my structure is like this way. So I should superimpose and I try to find out which are the common regions present. I'll make a map. Here you can able to see the maps, right? This region, this region, this region, this region. So such kind of maps I should make it out. Once I make it out, I can able to go as a blueprint and I'll upload into the databases. Zinc Mapper is there, Zinc Farmer is there. So many query databases are there. So it will give me look-alike structures. With this property, these are all the compounds I have in my database. Please take it. So that's the pharmacophore modeling role. So that will be discussed by Mazin in the next slide. So here are some pharmacist, format, these are the various tools which have been used. Next one is QSAR, quantitative structural activity relationship. As a chemistry point of view, you might have studied about the SAR. SAR is the first generation, where my structure will be present. Here I need then a donor group or acceptor group, or what are the thing, or electron withdrawing group, electron acceptor group, what are the thing may be. I will be reading the SAR, right? I have in this position it is required, in this position it is not required, activities increase. SAR. QSAR, quantitative. Every structure will have different properties. That based on the property, it will try to find out which are the properties playing a major role, so that for my designed compounds or the compounds which I have, I can able to predict the activity. I have a set of compounds which maybe it's a benzimidazole derivative. I have designed. I am not interested in doing out the molecular docking studies. Directly, I want to go for to know how much amount will be required, because everything depends upon the dosing. Because some drugs, if you see 50 mg, some drugs will be 500 mg. So based on that, if I want to work on, I need to do a study called as QSR. From that study, what I'll try to get it is which are the properties playing a major role. So I have my chemical structures. I'll calculate the properties and I'll submit into the equation. Biological activity is F of physiochemical properties. So this, it will give me the outline, how much amount this chemical structure will be required as a IC50 value or inhibitory concentration, whatever the thing I can able to predict. So if you look for any chemical structure, what are the various descriptors we can generate in the sense geometrical, thermodynamic, electronic, or constitutional, and topological. Various properties can be calculated. That property will be X 1, 2, 3, 4, 5. And as I said, right, some 30 compounds already marketed. So those compounds I'll be taking, or someone synthesis. Their value will be there. Y, I'll be saying as a Y response, X versus Y. So I'll be correlating Y, what is the thing to the X1, Y, what is the thing to the X2, X3. Based on that, which property is playing a major role in producing the biological activity, I'll choose and make the equation with some constant. With that equation, I can able to predict my activity score. So this in detail, I'll be explaining as a ML model development. I'll be this is the thing. And then the virtual screening, which will be playing a major role, where this will require a protein structure identification, protein structure preparation, ligand structure identifications, ligand structure, what do we call, preparations, molecular docking, result analysis, ADME, all together it will be covering up. So here, this will be discussed in the upcoming sessions. Proteins selection and molecular docking all will be discussed. So here, what we'll be doing in the sense, simple way, I have a identify a target. This is, so previously I did QSAR, right? After that I did pharmacophore modeling, right? All together, next step I should go on whether my drug will produce activity or not. That I should identify. No. For that I'll be carrying out the virtual screening, molecular docking studies. How effectively a drug will be binding to the receptor, that we will be predicting. So that plays a role. Simple example, I will say I have fall down. Inflammation is a disease condition. What happens? Because we know we have studied the pathway called as prostaglandins, arachidonic acid somewhere in 19%. Try to recollect that. So arachidonic acid pathway will be playing a role where it will be activating cyclooxygenase pathway, lipoxygenase pathway, COX-1 and COX-2, my inflammation. If I take celecoxib, it has to go bind with the celecoxib and suppress the inflammation. This is the mechanism happens, right? Same way, I'll be testing whether the synthesis, I'm going to synthesize some compounds, that structure, or I'm going to do a plant biology. I have taken a plant extractor. In that extract, what are the compounds I can able to do? So what are the things I can able to predict whether it can bind or not? For that, it can be utilized this virtual screening. This also will be discussing in detail. So in general outline, I'll be giving. So how effectively a protein drug binds to the receptor? You can able to see this image, how it's going and binding. As I said, 3D structure is playing a major role. The protein structure will also be in 3D nature, and the ligand structure will also be in 3D nature. So that only it can able to, you can able to see the bend and flexibility, all those things and all. So that plays a role. So this how a molecular docking will be carried out. And as I said, right, the ADME plays an important role. Why? Because many of the drugs which have been failed in the market is because of pharmacokinetic property. 39% of the drugs till today failed in the market only because of poor pharmacokinetic property. Rather than that, if you look into that, animal toxicity will be there, adverse effect in man, commercial reasons, lack of efficacy, that also plays a role. But most widely due to the pharmacokinetic property. Why? If I need a drug, if I'm taking a drug, that should go and attack a particular organ or particular site. If it is not reaching, without reaching the particular site of mechanism, if it get metabolized and excreted, there is no value for the drug. Whatever the drug I'm taking, it should go produce the biological activity. If it is not doing, there is no use. So here we will be predicting the ADMET, absorption property, distribution, metabolism, excretion, toxicity. So all together in detail, Ms. Naina will be explaining the next slides. So the softwares which we can use is PKCSM and Swiss ADME studies. So now I come to the next level of a study which is called as molecular dynamic simulations. So as you say, right, how done homology modeling, protein I have predicted, or is protein selection I have done, ligands I selected from a plant sources or the synthetic sources I have done, or pharmacophore from the databases I have collected, QSAR modeling I have collected. I have the structures and this thing, and I went for a molecular docking studies. I have done docking studies. Is it sufficient? No. You have to go to the next level. Why? Because molecular docking will take place in the empty space, vacuum. There is no disturbance in the other. But our body is having a water molecule will be present. I have a pressure bar, normal pressure, not my BP. In general, body will be having a pressure 1.0143 something it will come. That one, and my body will be having isotonicity. My body will be having a salt nature, NaCl will be present, right? All together it should be made, and in that condition, how it will be working out. For that, we'll be using a study called as molecular dynamic simulation. This study in detail, Mr. Srinivas will be explaining in the last session of our workshop. So what happens here in the sense, my body, how it will be, I'll be building the box, I'll be putting into that thing, and how it is playing with the drug and the receptor, how it is playing inside the body. It's just like a roller coaster, you can able to see, right? If I hold properly in a roller coaster, I won't fall down. If I don't have any grip, that holding, I said, right, interaction, those things will be explaining the future slides. So it can be of various things. It can be of what do we call? Membrane protein will be there, normal proteins will be there, receptors will be present. All together you can able to do. And to run this thing till the molecular docking studies, everyone can able to run all the tools in your own soft systems. And will be providing, as I said, right, almost ebook study will be sharing to your personal IDs. What are the presentation you are doing? including all together will be shared to your IDs. So you can able to download the software, or virtually if you want to run online also you can run till what do we call molecular docking result analysis. After that, to run this MD simulation, you need a high-end system where it requires GPU, graphical processing unit is much required to carry out those things. Rather than that, you can able to perform all. And moreover, some Google Colab is giving free, but we couldn't able to make it out. It takes a long time process. But rather than that, you can practice all those things. And so softwares used are all the softwares for this are free. Gromax, Desmad, Lance, NAMD, and those things and all. So I should prove, right? I'm saying computational practice, you're replying that these things and all. Now, is there any proof? Yes, we have the proofs. So dorosolamide, which is an carbon anhydrase, have been marketed through the help of AI or a computational tool, second and then Relenza. So these are the drugs have been identified, and some drugs have been repurposed. Repurposing also plays a role where computational tools you can able to utilize. So repurposing, when with hypothesis, I took some set of molecules for a new disease and I can able to try. So in such a way, we have got haloperidol as a lead compound for the non-peptide HIV inhibition. So such kind of things you can able to make it out with the help of computers. So what are the prerequisites for not only molecular docking studies, for the whole? The prerequisites required will be, first you should have a proper hypothesis. What type of work I'm going to do on? Either I'm going to work with a structure-based drug design, ligand-based drug design, fragment-based drug design. First, I should have the clarity. Second, okay, if I'm going to work with a structure-based drug design sense, I should have a proper protein. If you are not taking a proper protein, it's of no use. Selection of proper protein plays a major role. When the protein is selected, then how I have to make it out? What are the steps involved? That will be discussed. Ligand structures, chemical structures. Because as a synthetic chemist, I have designed some components I'll be working on. As a pharmacologist or pharmacologist, I'll be doing something also. Same way, each and everyone can work with any ideal. But the chemical structures I should download, where I should download, how I should convert, those things will be discussed. So when that is done, I should carry out the molecular docking studies. How to perform? So many people say that I have done molecular docking studies, I have not got the results in a proper way. How to overcome? Whether my results are right or wrong? Yes, your results will be right only if you follow the stringent protocol. If you are not following the stringent protocol, what are the results you are going to get? It's of no use, because the protocol which you are going to follow will give you the best output. How fast, how far I'm utilizing the various tools to analyze the results, analyze the data, then only you can able to make it out. So based on these things, if I work out, I can able to make it out a better compound or better product for the next process and all. So this how you have to create the hypothesis, the sources, and we'll be sharing all the links for the softwares. Is there any questions? Any online questions? No questions. Yes, ma'am.
Uh, just for the auto docking, what are the criteria for the selection of the software? Because there are multiple softwares. Okay, so which software will show the best results? Means we have Autodoc Vina, we have Gold, but what is the proof? Means which software results are best? On what basis we can? Because there will be difference, obviously.
Everything is an algorithm. Example, by seeing this fellow, I will give something. I'll have an impression. That is my algorithm. Okay, this fellow looks like a good, this fellow looks like a bad. My calculation. Same you will be having, right? That's the difference. Point number one. Point number two, all the software they are utilizing the wave equation. Stored in this wave equation as the value, mathematical equation will make it out. Second thing. Third thing is, for which criteria I'm giving how much weightage. That makes the difference. Example, for some targets or some working on that. For covalent bonding, I may give more weightage. you may give less weightage. So that the difference may happen. And the next one is maximum of the softwares which are paid and some are free. In the freeware, I always prefer Autodoc. Autodoc plays a major because it's from the Scripes Institute. They are giving from a longer. Autodoc and Autodoc, there is not much difference with their scripts. Only the difference is Autodoc, one versus one protein, one ligand. But Autodoc, one protein, multiple ligand you can able to work on. So that's the only difference you can able to see. Rather than the score-wise, no issues. As I said, right, the pre-preparation process, how we are going to make out of which protein I have to select, what should be its resolution, whether I have selected the right packet, all those things. And after getting the result, I should analyze the result in proper way. Sometimes, example, they will say -8 and -10 is the score. Compound 1 showed -10, compound 2 showed -9, compound 3 showed -8 as a result, output score. So once it is showed, I should not blindly go with -10. That's wrong. Why? Because already my test compounds or the compounds which are marketed may bound to some amino acid. That same thing, whether it may test compound retained or not, that I have to predict. If the compound 2 have retained, that means out of my 10, -10, -9, -8, no, -9 plays a major. That if you take and go for further studies, you get a proper result. If not, you won't get the result. That's the differences. I have published a paper in Nature with Autodoc Vina only. All pre-research only. Ma'am, it all depends upon the hypothesis, how we are going to convey the science. The authors, how by reading that the reviewers, okay, something I've done and that. So it all depends. Again and again, I'm stressing the hypothesis, right? That matters a lot. So you can able to do publishing everywhere. I said as a scientist, Nature is the highest of journals, right? So in such a journal, they are accepted, means it is accepted. The protocol, what we have followed, it should be convinced to the reviewers. Thank you. Any other questions? No.
Any other queries to be asked?
Any online queries? Oh, Mike.
Sir, you told us about the docking score now. And we have different docking interaction like hydrogen bond, hydrophobic interaction, ionic interaction. Okay, so actually, when we select the docking score, whether we have to think about the interactions.
Definitely, we have to think about the interactions.
Okay, so actually, on what is the weightage of
hydrogen bond will be the first. Hydrogen bond will be the first. And then rest and all will be in equivalent. Why I'll say? As I said, the molecular dynamic simulation. Molecular dynamic simulation, I said right. This docking will indirectly affect in molecular dynamic simulation. Example, if I have a compound, I did a docking, my score is -10. Point number 1. Point number 2, it is not at all having hydrogen bond interaction. No hydrogen bond, only hydrophobic or ionic or pi stacking is there. Third, if I do a dynamic study, the compound won't be stable. It won't sit into the packet because I don't have the stable connection. So that's the reason. So in case if my score is also less, if it is ready to pack within that, then that can be considered. So you should always look for the hydrogen bond donors. Sorry, hydrogen bond interactions only. That plays a major. Dynamic simulation, I said right. This is how it will affect it. If I have a chair, a rolling chair, only or not a simple stool will be there. If I'm sitting, if you try to rotate me like anything, what happens? Either I'll fall down or I'll break the stool. That means the protein and the ligand, what it bound, it will break out. But the same way, if I'm sitting a chair where I have the hold, if I'm holding, that holding is called as interaction, hydrogen bond interaction. If I have the holding, if you rotate left, right, what are the 360 degree, I'm having the hold so that I'll sit into the right packet. So that's why here this will affect that. So always look for the hydrogen bond interaction. That will be the more important things.
Okay, sir, after getting the docking results, okay, it is a kind of in vitro or in vitro studies. Okay, we have done the docking using Autodock software, but after getting the good results with the software, okay, all the parameters are good. So we should proceed with the in vivo. Okay, but we are getting the opposite result. Is it possibility?
Chances are 50-50%. Point number 1. Why? Because some drugs which are potent in vitro failed in vivo. Some drugs which are potent in vivo fail in vitro, right? Here the same way. Why? Because the biological system has to support what you are taking the drug. It will go for a biotransformation.
Yeah.
Some drugs will act like a prodrug. After reaching the body, with the help of enzymes, it will get converted and will produce activity. That we can't able. 50-50 only I can able to give the choices. Because body is a complicated system, right? If he has to reach the cycle before reaching the site, if it failed, that also plays a role. So that's why we have to follow the stringent protocols. Then you can get this. Yes, we have done so many studies, as you said, right? We have done with anti-cancer, repurposing we done, we have done Alzheimer's, all the results we got good only. So once you follow the protocol, you can get it, ma'am. That's it.
I have a small question. Like when we do the docking, we get for one single molecule, we get multiple scores. The one with the minus negative, the highest negative score will be to choose that one. So when we do the same molecule for visualization purpose, when we do 3D interaction and all, the interactions, if we see there, we get multiple conformation changes for the same docking. So how do we choose? Sometimes for like
Which tool you are speaking?
normal Autodock Vina for docking, and for visualization BioWare Discovery Studio, because
Fine.
it's more suitable. So when I do that, what happens for the one with the highest score, -9, it is sitting in the pocket. When I go to the next model number 2, same molecule, it is somewhere out of the pocket or a different binding site. So on what basis can I proceed with this molecule?
Yes. So docking will more detail they'll be discussing tomorrow, but in general I'll be giving the preamble. Docking will have 2 parameters. One is search algorithm, another one is scoring. Search algorithm sense, example, this is my packet, active packet. I'll consider this is my drug. What happens here? It will try to bind in this position, this position, this position, various positions. This is called as trying to bind. And once it is done, it will produce a score. Second thing, and where I'm going to sit as a comfortable, the score will be more. One thing, because of that one day, and you can able to see the scores. If you look for the same compound, you can able to see RMSD, LB, RMSD, UB. That means how comfortable. RMSD is a root mean square deviation. The protein structure is here. Once the drug bound, is there any deviation in the nature? If it is deviated, this drug is pricking, maybe a poisonous one, maybe a toxic one. So we should not consider this pose. So all the thing, if you see the thing, RMSG only for a top score, it will be giving 0, rest and all, some values will be present. That's the reason we have to cut down those things. So anyway, tomorrow's session, they will be discussing in detail about the molecular things. Thank you. I'm better change ಮಾಡಿದ್ಯಾ ಬ್ಯಾಟ್ರಿ ಚೇಂಜ್ ಮಾಡಮ್ಮ ಅದರಲ್ಲಿದೆ ನೋಡು ಬ್ಯಾಟ್ರಿ ನಿಮಗೆ ಅವ್ರ ತಗೊಟ್ಟು
Many things as different tools you discussed. What is my being a pharmaceutics and pharmacology person, my in all these sessions that I want to clear now, that is when one ligand candidate will have the interaction with some ligands, we will search to binding things and binding affinity. So when comparing the scores, we compare the binding things and binding affinity. Now, binding affinity also depends on the biological system when it will enter into the body. Now binding affinity and binding score, if you will compare that, which software you will be preferring, docking or MD stimulations?
The MD simulations.
MD stimulation.
Ma'am, docking is the preliminary steps. MD simulation plays a major role in confirming, because as I can give an example of our one study only. We did drug repurposing using ML models and all. We found out some top 25 compounds, top 100 compounds with the scores. I did for docking. From the docking, top 20 compounds I took. And then after analyzing 10 compounds I took for dynamics. Out of the 10, 5 I took for in vitro. Failed. Only one passed. That we took for in vivo. As you said, right, MD simulation gives you one by one. Because only one drug, recently we published a paper on capsaicin for a cancer study. So that with almost some 20 receptors we did docking. Then only to one receptor it bound properly with 1,000 nanosecond after simulation also it retained its length. That showed a better results in vitro and in vivo. So MD simulation only will give you the best result, not the docking.
Not the docking. Thank you.
Yes, sir. Generally, CADD is hypothesis, right, sir? Basically, my question is like, how much we can rely on CAD, sir? Because CAD is based on computational method, basically mathematics. So it is how we'll generate a 3D structure of molecules like amino acids. Not necessarily they should bind or like no, but some amino acid may have ability to bind. Have hydrogen bonding, sir. But when they are in protein structure, they may not process hydrogen bonding. But how much extent that a computer can simulate it, sir? Like how much we can rely on CAD?
So computational study is like an astrology. Astrology, you know, right? If you know the right astrologer, you'll get the right results. Because every street we are seeing astrologers. Next year will be super 2024. How it went? So it says that you have not met the right astrologer. One thing. Second thing, what I say is protocols. You can rely on computation tools. It's not a wrong thing. How far we are going to use that thing in a best way. To use that thing, okay, I want to perform one sentence. You can cross validate. People, what we are doing is we are doing document finished up. Validation we will do. I'm not validating the same tool. If I do the validation, cross validation, then only I'll come to know, okay, this method is right. Then you can able to relay. Computational tools, you can relay 50-50 only. In vivo, again the same questions. As the drugs, some drugs which are potent in vitro are failed in vivo, which never showed activity in vitro, showed best activity in vivo. Because our biological system, one thing, second, what type of chemical structure we are taking. In case we are having the what we call COR group, ester group, such kind of things, and this, our body having of more of esterases, where it will break down as a proteolysis process, and it will break down and it will give the product. Maybe that metabolite may be active, may not be active.
But by doing CAD, we may leave those compounds which were potentially active. But
chances are there. I agree. Chances are there. There is no doubt. I agree.
Basically, it is hypothesis.
Yes, that's why we have now. We are rather than that, we are going to introduce one more tool called as network pharmacy. We will be discussing in the next session. So if you take a plant, I said right, a plant will be having some 30-50 compounds. All 30-50 compounds may not be going to produce activity. Some will be what we call long chain fatty acids will be present. That is of no is only to improvise the thing only. So we'll be taking only the core active constant and we'll be working on that. That we will be discussing. There you can able to get much more idea.
Sir, in plant constituents also, Lipinski they won't follow.
Lipinski rule I won't
because of
follow.
their size,
No.
molecular weight.
Lipinski rule is a rule. It's a hypothesis. All the rules are you obeying?
Active component we cannot predict, no, sir.
That's
through
what
CAD.
I'll also all the rules are we obeying?
No, sir.
No. Lipinski is a scientist. What he did is he created a hypothesis. It's also his hypothesis. He took some compounds and he made a average weight of how much it is this. So based on that, he gave a hypothesis molecular weight, donor acceptor. Do you know the last year what is the molecules marketed? Previously, last year, the average molecular weight of the compounds which is marketed is 250 to 300. Now 400. Drugs are became obese. So it is not required to believe on this hypothesis. Second thing I'll say, for a tuberculosis, there is a drug where its molecular weight is more than 2000 Dalton. It is working right. He gave his hypothesis. If you are interested, you take. If not, leave. That's the thing. Thank you.